Hello, hello, hello. Good morning, good afternoon, good evening, wherever you are. Welcome back to another session of DNA rejuvenation technology. Today is the seventh video and it's a celebration. Welcome, celebration. Celebrate the dance, come on. Okay, today is a celebration. It's the seventh video. We are going to start the last stages of the production, which is the purification. And today's topic is purification of EVOSK virus, tangential flow filtration. Okay, in other words, purification means just getting rid of the contaminants we have produced EVOSK, and we want this is EVOSK, right? And you see the little dots and things that are not EVOSK. So what we are going to do is getting rid of whatever it is not EVOSK. That is in plain words of what purification means. Okay? So we are going to get rid of the contaminations. And at the end of these stages, we are going to get clean EVOSK ready to use. The therapy, the magic potion, EV with OSK. Yay! Okay, in this occasion, in order to purify, in order to get rid of the contaminants, we are going to use a technique that is called tangential flow filtration. It sounds funny, but in fact, it's just using pipes with filters, and these filters separates the EV from the contamination. These filters are big enough to get rid of the contaminants, but small enough to retain the EVOs. That is the main principle of the tangential flow filtration. Very, very easy. We just get rid of the contaminations by using filters. Okay? Easy peasy. Let's go for it. Let's go for today's topic, purification of EVOS virus using tangential flow filtration, the seventh video. Let's go for it. And before starting, we are going to give a little introduction. In case you have just landed here, you need to know what we are doing. And yeah, this slide is a reminder of the technology. Okay, so we are going to see what we saw in previous videos. OSK reverse the age of the DNA. Okay, do you remember the DNA? This little piece of information that have all the information that you need to build blocks in your body. Yeah, do you see the DNA here? That is correct. This has all the information that you need for doing everything, pretty much everything in your body. Do you see here the little dots? Yeah, these are marks, little marks. And these little marks are patterns. Okay? These indicate to your cells how to use the DNA. So this is extra information. Do you see here these beautiful young people? Yeah, these are typical marks in young DNA. So you only have a few marks, and the DNA is easily accessible. When you are young, you have a little amount of marks, and the DNA is easily accessible. But as the time pass, your DNA start piling up a lot of marks, as you see here, right? As you see here. These little marks are extra information. This extra information sometimes said, don't read DNA. In this occasion, regarding these genes, this extra information It's telling your body not to read the DNA. Okay? So this is a typical old pattern in DNA. You have a lot of these little marks, these little stamps. And these little stamps, as I have said, are telling to your body not to read DNA, so your cells lose functionality. In other words, you get older. Oh, but we have, remember, OSK, the magic potion. The magic potion. You see here OSK with the DNA. Okay, OSK, the magic potion, is able to get rid of the marks. Yeah, it cleans DNA from having so many marks. In other words, your DNA reverts to this state in which it has only a few marks, and your DNA is accessible again. Your cells can read the DNA again. Your cells rejuvenate. You rejuvenate. You go back in time. You recover youth. You are young again. Okay? Happy? Oh, yeah. Yeah. Let's go for another slide. Let's remember a couple of things that we need to remember before uh, resuming. Let's go for it. Okay, let's remember the production of AAV-OSK. The three main steps, the three main stages. Okay, very, 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 very easy. We already saw it in previous videos. Okay, so remember, first of all, we need the DNA, the three DNAs. The DNA with the OSK genes, the DNA with the red cap, that is the DNA to produce the virus capsid, let's say the box, and then we need a third DNA that is called helper DNA. The helper DNA will help to include the OS genes inside the EV particles. Okay, so okay, let's go for the first step. Once we have our DNA designed and we have ordered it, the first step is producing millions of copies of DNA. And the only thing that we do is introducing the DNA inside bacteria cells. We introduce the DNA inside bacteria cells and they will replicate. They will grow in number. And as they grow in number, they will duplicate the DNA. So by growing and duplicating the DNA, at the end of this step, we will have millions of copies of DNA, as you see here, right? A lot of copies of DNA of each single, if each is single DNA. Then we break down the cells and we get the DNA. Okay. So once we have a lot of DNA, we can go to the second step. Okay, the DNA OSK is expanded and encapsulated inside EV. This is the second step. So once we have our DNA, we use another type of cells, mammalian cells, to produce EV particles, as you see here, and the cells include the DNA in the specifically the OS genes inside the EV particles. This is the second step. Is just introducing the DNA that we have produced inside these cells. Okay, and the cell will do all the work. The cell will do all the hard work, which is producing the EV particles and including the OS genes inside the EV particles. Okay, that is the second step. Producing EV particles with the OS genes inside. And once we have finished this step, it's just the following thing, the third step, which is cleaning the AV particles with us. Okay, and this is today's topic, cleaning, purifying EV particles using tangential flow filtration. Easy peasy, right? So we have the DNA, we produce millions of copies of the DNA, we introduce this DNA in special cells, mammalian cells, and these cells produce EV particles, viral particles, and include the OS genes inside the EV particles. Third step, purifying the EV particles with the OS genes and leaving these ready to dance, ready to rejuvenate ourselves, ready to rejuvenate us. So let's go for the next slide. It's going to be a little bit introduction about tangential flow filtration. It's very easy. Remember, it's just a fancy filter, okay? Just a fancy filter that separates the EV particles from the rest of the contamination produced in the production of AAV-OSK. Let's go for it. Hey, let me introduce you to the tangential flow filtration technique, TFF. TFF? Yes, it's a very easy technique. Look, the actual device looks like this. Okay, it's just a system of pipes. You see here, this is the inlet pipe. The sample enters this way and it goes through the device that it's just a system of pipes, and it goes out through this pipe, the outlet pipe. Okay, this is a representation. The sample enters this way and it goes out through this pipe, the outlet pipe. Okay, okay, the principles behind these techniques are easy. Let's read with me. The sample runs through the duct, through the pipe of the TFF device, and the walls of the duct are porous. You see here? This is a representation of a pipe, and the walls of the pipe, one side maybe is porous. It's kind of like a membrane. You see here the porous. You see here all the porous. Okay, so the walls are porous. So the solvent, let's say the buffer, and components that are smaller than 500 kD, well, this is just a measurement. The solvent and the components that are smaller than this size escape the duct, okay? They escape. You see here, these smaller components, these components and the solvent, the buffer, are smaller than the porous, so they escape. You see here, they escape. They escape, they escape, they escape. So it has two effects. First, it concentrates the sample because you reduce the amounts of solvent, and it purifies the sample because you get rid of the contaminants here. You see here? You see you. So there are two effects. First of all, concentrating the sample. Second effect is purifying the sample. Okay, what happened with all this solvent and all these contaminants? They go through this third pipe. Okay, the third pipe would be, let's say, here, like whoop, like doing kind of something like this. Okay, so they would leave the sample through this pipe. Okay, this other outlet pipe, this waste pipe. So this is the waste pipe. That would be the waste. Okay, and what happened with the EV-OSK? Which what happened with EV-OSK is that it's bigger than the pores. You see here, the EV-OSK is bigger than the pores, so the pipes retain EV-OSK. Okay, so at the end of the process, you have EV-OSK concentrated and clean from contaminants. Okay, so just this technique is as easy as that. It's just using pores to get rid of the pores that are bigger than the contaminants and the buffer. So you get rid of buffer and contaminants, and at the end of the process, you have EVOs clean and concentrated. Okay, let's go for the next slide. Oh yeah, so here we are in the next slide. In here, we are going to see how the whole system works. Okay, how the TFF is integrated in the whole system. Okay, and the system comprises of two circuits. Okay, a closed circuit and an open circuit. Let's see both of them. Okay, the sample will run, let's read with me. Okay, so the sample will run through a closed circuit and it will go from a sample container. You see here, this is the sample container. It contains the sample, and as you can see here, the sample has some contaminants, okay, from the expression. From the sample container, it will go through this pipe to the pump, okay, here, the number 2. The pump pumps the sample, okay, through the system, through the rest of the system of the circuit of the closed system, circuit. Okay, so the sample now is here in the pump. The pump is pumping and goes through this pipe. And it ends up in the TFF device. Okay, it will enter the TFF device through the inlet pipe. It will go this way, this way, this way, this way. It will cross the TFF device. It will go out the TFF device through the outlet pipe, and it will end up in the sample container again. So as I have said, this is a closed circuit. Let's let's go together, okay? This, through the circuit again. Sample container, pump, TFF device, and again. This is a closed circuit, okay? What happened regarding the other circuit? The other circuit, remember, is an open circuit. It starts in the oh 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 in the waste pipe, that is this one here, okay, and it's represented here, the waste pipe. And it ends up in the waste deposit, okay, as easy as that. So the contaminants and part of the buffer will go through this system. The contaminants and the buffer cross the pores of the TFF and they are deposited in the waste deposit. Easy, right? Okay. So this is the whole system, and let's see what happens when we turn on the machine. Okay, let's see what happens. You already know, but it's good to go through it together. So let's look at here. Uh, okay, so remember you have here the sample with some of the contaminants, as you can see here. It goes from the sample deposit to the pump, from the pump to the TFF device. It enters in the TFF device through the inlet pipe. And in here, you will see here the sample with some of the contaminants enter in the TFF device. And as it crosses the TFF device in here, the contaminants and part of the buffer that are very small cross the TFF pores, the TFF membrane. You see here, right? So the contaminants and part of the buffer go out the TFF device through the waste pipe, okay, through here. So as you can see here we have contaminants and a lot of buffer, and it ends up in the waste deposit, okay, while the AVV-OSK is retained in the system. Why? Because as I told you, EVOS is bigger than the pore, so it cannot cross the pores, okay? So the EV-OSK goes out the TFF device very clean and concentrated throughout through the outlet pipe. Okay, as you can see here, this is the outlet pipe, so it ends up here. This cycle is repeated many times, once and again, and as it happens, you see as EVOs is cleaning more and more and more and more, and as it is clean, at the same time it is concentrated. So at the end of the process, you have here EVOs very clean and very concentrated. The volume is less, is smaller. Okay, so at the end of the process, we clean and concentrate EVOs, and EVOs now is ready to work, ready to help us, ready to rejuvenate us. This is almost the final product. Happy? Very happy? Oh yeah. So now what we have left is just seeing how to perform this technique step by step. Okay, let's go for it. Let's enjoy. Well, here we are in the steps towards purifying the EV-OSK. Okay, and there are only 3 steps. That is correct, Mr. Sumer, Mr. Novo Sumer. Only 3 steps. Okay, let's go for them. The first step is, let's read with me, okay? Thaw the expression supernatant if it was frozen, okay? Centrifuge it and incubate it with benzonase and magnesium chloride for 30 minutes, and afterwards incubate it again with some detergents, okay? Let's understand this step. Okay, so in the first step, we centrifuge the sample. It's because there may be some cells from the expression, so we want to get rid of some cells by the centrifuging. Okay, and we add benzonase and magnesium chloride. Why? Because the sample will be a little bit contaminated, so with DNA. So the benzonate will break down the DNA, will break the DNA. So we will get rid of the DNA with this enzyme that is called benzonase. And the magnesium chloride stabilizes the DNA. So by stabilizing the DNA, we isolate the DNA from the AV-OSK. Okay, so we incubate for 30 minutes, and afterwards we add detergents. The detergents are able to isolate the AV-OSK from some of the contaminants because the detergents stabilizes the AV-OSK. Okay, so this is the first step, just centrifuging and incubating first with benzonase for 30 minutes and afterwards incubating with detergents. Okay, well, the second step is preparing the TFF buffer. Okay, is preparing the TFF buffer and equilibrating the TFF with the buffer. That is to say that we want to prepare the system to receive the EVOs in the best conditions. And the best conditions is this buffer that we are going to prepare. Okay, so we just circulate the buffer through the system, and the system will be equilibrated with this buffer. This is the second step. As you see here, we will put here the buffer, and the buffer will go to the pump to the TFF, from the TFF to the recipient of the buffer. Okay, and in the open circuit, you see here, the open circuit will be also connected to the buffer recipient. So that will go this way in a closed circuit, and also in this occasion, the other circuit, the open circuit, will be closed because, as you can see here, the buffer will run through the waste pipe to here again, so everything will be closed. What is the third step? Okay, the third step is once the TFF is ready, we set up the circuit. As you can see here, we set up the circuit properly. I will show you how, and afterwards we will just turn it on so that the sample will be more concentrated, cleaner, and we will also buffer exchange the protein. We will add a little bit of excess of buffer, TFF buffer, in order to change the media of the AV-OSK. So at the end of the process, we will get AV-OSK clean, concentrated, and it will be in another buffer, in the TFF buffer. Okay, these are the three steps. First one is centrifuging the sample and incubating with Benzonase, and afterwards with detergent. The second step is preparing the TFF system, and the third step will be concentrating, cleaning, and buffer exchanging the AV-OSK. You see here the protein, this is AV-OSK, okay? So let's go for it. Let's go step by step. Only 3 steps. Wow. Okay, let's see the first step. Okay, the first step is centrifugation and incubation with additives, as I told you, okay, in the steps in the previous slide. So what we have here, okay, let's read with me. To, it's frozen, the expression supernatant, centrifuged and incubated with benzonase magnesium chloride for 30 minutes, and afterwards another 30 minutes with the detergents. Okay, so let's go step by step. Okay, in the first step, as you can see here, we do the expression supernatant if it's frozen. Okay, so we leave the supernatant several hours at room temperature until it's completely thaw, and we are checking the temperature so that the temperature doesn't excess go farther than 4 Celsius degrees. Okay, we want the sample nicely cool. Okay, this is the first sub step. Afterwards, what are we going to do? We centrifuge the sample. We centrifuge it in a centrifuge device at 7,000 Gs for 15 minutes and 4 Celsius degrees. By centrifuging the sample, what we are doing is getting rid of some cells. There will be not many cells, to be honest. The supernatant is very clean, but just in case we have this step. So at the end of the process, we will have the supernatant, the remaining cells in the bottom. So we will just transfer the supernatant to a clean recipient like this, and we will get rid of the cells. We don't want the cells, okay? We treat the cells here as contaminants. Let's go for the next super step. That is very, very easy. We just add Benzonase, okay, at this concentration, 10 units per milliliter. Okay, you can calculate it. Just have in mind that you add the Benzonase and the magnesium chloride to the supernatant and incubate for 30 minutes. We do it to get rid of the DNA from the cell. The DNA of the cell is kind of a contaminant, okay? This is DNA from the cell. We don't want this DNA. So we add Benzonase, which is an enzyme, as I told you, that breaks the DNA. So by incubating 30 minutes with the Benzonase and the magnesium chloride, we get rid of the DNA. You see here, after incubating, it will look like something similar to this. We will get rid of the DNA. These are some other contaminants, okay? And this is the DNA as a contaminant. So the magnesium chloride, what it does is stabilizing the DNA. So the DNA will tend to dissociate from the AV-OSK. And the Benzonase, what it does is cutting the DNA. So at the end of this process, we will have allegedly, of course, EVOs free from DNA or dissociated from the DNA. Okay, so let's go for the final sub-step in this step, which is adding detergents. These detergents are chaps at 0.2% and the oxycolate at 0.1%. Okay, these are detergents. And what happens with these detergents? Okay, one of the properties of the EVOs is that in the surface may be a little bit hydrophobic. So by adding detergents, we're kind of stabilizing the EVOs. So by stabilizing the EVOs, we avoid certain contaminants from interacting with EVOSK. So by adding detergents, what we are doing is separating the EVOSK from some of the contaminants. So as you can see here, after an incubation of 30 minutes, you have on one side the EVOSK and on the other side the contaminants. EVOs will be here interacting with the detergent and the contaminants will be here. Okay, so this is this first step. Yes, thawing, centrifuging, incubating with Benzonase to get rid of the DNA, and incubating with the detergent to separate the EVOs from the contaminants, from some of the contaminants. Okay, so easy peasy. This is the first step. Let's go for the next step. Okay, so in the second step, a very easy step is just equilibrating the TFF with the buffer. Okay, so the first step is just preparing the buffer. I leave the ingredients here. You can read them whenever you want. I will just mention, okay, but the components are here for you. Whenever you need them, you can resort to this. So the buffer will be 50 millimolar of sodium phosphate. Okay, so you just add 50 millimolar of sodium phosphate, 100 millimolar of sodium chloride, salt, 1 millimolar of magnesium chloride. Okay, 1% of glycerol, and then chaps at 0.2% and the oxycolate at 0.1%. Okay, let's break it down a little bit. The pH 7.2 is similar to the pH of the cells, and also kind of similar to the pH of the blood. That is why we are using this pH, and also because EVOS is happy in this pH. Then the sodium phosphate will be the buffer that let's say that buffers the sample. Okay, the salt is because we always need a little bit of salt that equilibrates whatever protein you are working with. Also the EVOS is happy with salt. The magnesium chloride, because just in case there is a little bit of DNA, the sodium chloride will stabilize the DNA. By stabilizing the DNA, we're avoiding interactions between the DNA as a contaminant and the EVOS. And the glycerol, why? Because the glycerol works very good sometimes when you have a protein, or in this case EVOS, with a mixture of hydrophobicity, some polar amino acids, that works very good, the glycerol. Okay, it stabilizes the EVOs. By stabilizing the EVOs, we prevent it from interacting with other contaminants. And the chaps and the oxycolate, as I told you, they were detergents. Okay, they will help us to isolate the EVOs. That is why each component, each one of them, have a purpose. Okay, so what are we going to do? We set up the TFF system. All the the things will be connected to the buffer recipient. Okay, so let's read with me. The waste pipe, this pipe, okay, you remember, will be connected to the buffer recipient. Also, the outlet pipe, this pipe, will be connected to the buffer recipient. Yeah, also the inlet pipe will be connected to the buffer recipient. So everything will be in a closed system. So the buffer that will be here will go to the pump. From the pump, it will go to the TFF system. From the TFF system, it will go to two ways. It can go either to the waste pipe or to the outlet pipe. Both of them will end up in the buffer container. So everything will be set up in order to get a closed circuit. Remember, that was the closed circuit and that was the open circuit. But in this case, since we want to get to clean or equilibrate the system with the buffer, we'll close both of them. Both circuits will be closed. I repeat, we will have the buffer container here. With the buffer, it will go to the pump, from the pump to the TFF, and from the TFF, it will take two ways. It can go either to the waste pipe, up to the buffer container, or it can go to the outlet pipe, up to the buffer container. So everything will be in a closed circuit, okay? And we will run the the system for 20 to 30 minutes, so that at the end of the process it will be clean and equilibrated with the buffer. So okay, after the setup, the system will be like this. Okay, the buffer here, the waste pipe here, the inlet pipe here, and the outlet pipe here. Okay, so after setting up the system, what we are going to do is turn it on the pump. So we turn on the pump, and what is going to happen? Let's see. As I have told you, the buffer will go from the buffer recipient to the pump, from the pump to the TFF. And inside the TFF, when you have an excess of pressure, it will go to the waste pipe. You see here? So the buffer will go to the waste pipe. It will end up to in the buffer container. The second option is that the two ways. The first way the first path I told you, the waste pipe, and the second pathway that it can take is the outlet pipe of the TFF that will end up in the buffer container as well. Okay, so we just circulate it, we turn it on the pump, and we leave it for 20 to 30 minutes. And after 30 minutes, we will have the system ready to receive the sample. Easy peasy, right? It's just preparing the buffer, setting up the TFF system, which is just only putting the inlet pipe, outlet pipe, and waste pipe in the buffer container. End of this task, nothing else. And the third step is just turning on the pump and leaving it for 30 minutes. So easy peasy. Buffer, putting the pipes where they should be, turn it on the pump and leaving it for 30 minutes. It can be done in 3, 5, 6, you do it in a blink of an eye. Okay, so let's go for the third step, which is very easy as well. Super easy. We will enjoy it. Ooh, the third and last step. We are at the end of the story. Okay, super easy step. The step number 3. Clean and concentrate the sample. Yeah. Oh yeah, Mr. Novosumer. We concentrate, we clean, and we buffer exchange the protein. Okay? So let's go for it. In this first step, there are 3 super steps. Okay, so let's go for the first one. Let's read with me. Set up the TFF pipes with the correct position. The inlet pipe and the outlet pipe both will be sunk in the sample container, and the waste pipe will be placed on the waste container. Let's see it, okay? In this occasion, we will have the sample here. You see here at the beginning, the sample is dirty. You see here the EVOs is dirty with contaminants, with some remaining with some contaminants. Okay, so it's here. So what we do in this step is the placing this pipe, the inlet pipe, and the outlet pipe in the sample. Okay, that is the closed circuit, as I told you. And the other pipe, the waste pipe, will be connected to the waste container. Okay, and what we are going to do is just regulating the pressure of the system here. Why? Because when you set up a pressure limit, whenever you turn on the pump and you have an excess of pressure, the excess of pressure what makes is that it creates extra pressure in the system, and this extra pressure will be used to get rid of some buffer that will come here, and with some of this buffer there will be contaminants. So by regulating this gauge, this pressure gauge, what we are doing is creating extra pressure. And by creating this extra pressure, what we are doing is that some of the sample, that is to say, some buffer and some contaminants will go to the waste pipe up to the waste container. Okay, that is why. So let's repeat it. The setup is as follows. We will have the inlet pipe and the outlet pipe of the TFF connected to the sample. And we will have the waste pipe connected to the waste container. And we will set the pressure limit up to 1.5 bars of pressure, okay? Yeah? Okay. So first step, setting up. Connected the inlet and outlet pipe to the sample, and connecting the waste pipe to the container. Afterwards, setting the max pressure of this gauge at the 1.5 bars, and setting up the speed of the pump up to 29, which is 2.5 liters per minute. Okay, this is the first sub step. Let's go for the second sub step. Is very, very, very easy. The only thing that we have to do in the second step, in fact, is turn it on the pump. Everything was setting up, was set up. So the only thing that we do is turning on the pump and leaving it until it works. That means until the volume reduces and until we get rid of the majority of the buffer and contaminants, as you saw. Okay, so we leave the system circulating until you have here less sample, until you see that all the volume is reduced up to 0.5 liters. So you leave it circulating until the sample is clean. Let's see how it works. Okay, let's remember it a little bit. So we will have the sample here. It was a little bit contaminated, as you see here, and it goes from the inlet pipe to the pump that is on, from the pump to the TFF. In the TFF, remember, you have inside the TFF a membrane, and this membrane has pores. The pores are small. Since the pores are small, the EVOs cannot go through the pores. They cannot go. So the only way that they can take is by going to the outlet pipe, again to the sample container. Okay, that is the circuit of the sample. So we are cleaning by this way the EVOs. And what happened in the second part that it can take? Remember, inside the TFF again you have this membrane that has a small pore. Okay, and when you create an excess of pressure here in the pressure gauge, when you create an excess of pressure, the contaminants that are very small cross the membrane. Okay, so they cross the membrane of the TFF and they go to the waste pipe. They go to the waste pipe to the waste container. So at the end of the process, what you are doing is losing some contaminants, losing some buffer. So at the end of the process, you have clean EVOSK, and you reduce the volume in which the EVOSK is, and also you get rid of this excess of buffer and the contaminants. So at the end of the process, you will see a kind of a small volume. So at the beginning it was a big volume, let's say 10 liters, 20 liters, whatever. At the end of the process, you have just 0.5 liters. So a very small volume of protein that is very, very clean. So at the end of the process, you will see a container with a small volume and the waste container with a big volume of buffer and contaminants. You see? Let's repeat it again and again and again until we understand it. At the beginning of the process, you have the sample here with the contaminants. We turn it on the pump, and the sample will go from the sample container to the pump, from the pump to the TFF. In the TFF, the protein that cannot pass the pores will go to the sample container again, and the excess of contaminants and buffer will go to the waste container. Okay, so at the end of the process, you will have a small volume of concentrated EVOs that is also very, very clean. Okay, understood this step? Okay, in the last step, once we see that the volume is reduced to 0.5 liters, we make the most of this circumstance and we add extra buffer, the TFF buffer that I mentioned in the second step. So when you see that this is reduced up to 0.5 liters of sample, we just add extra 2 liters of buffer. So we are exchanging the buffer. Now we have more of the TFF than the buffer or in which the EVOS was. Okay, so we add here 2 liters of buffer. So instead of having 0.5 liters of concentrated protein, now we will have 2.5 liters of protein. We continue circulating. Okay, so we will continue circulating again. By continue circulating, the sample will concentrate again. Okay, we will reduce again. So the excess of the buffer will end up here with the contaminants and with the other buffer. So at the end of this step, we will have clean protein. We circulate it until the volume of the sample is again 0.5 liters. So at the end of this process, you have first very clean protein, very, very clean protein, very, very clean in a small volume. That is to say that the protein, the EVOS is very concentrated. Plus, since we add the TFF buffer, we have changes the buffer. So at the end of the process, you have clean protein, concentrated protein, and in the buffer that you want. Super easy, super nice, easy peasy. Okay, so we repeat. We set up the system. We turn on the pump. That is the only thing that we do, and we wait until the sample volume is reduced up to 0.5 liters. End of the story. Nothing else. Once the sample volume is reduced to 0.5 liters, we add some 2 liters of buffer, We turn on the pump again and we wait until the volume is reduced to 0.5 liter. Easy peasy, right? And wow! Now we have clean, nice, concentrated EVOs in the buffer that we want. In a buffer in which it is very, very happy. Very, very happy. Yay! Yay! Okay, let's go for the final slide. Let's summarize everything so that you can keep it nicely in mind. Okay, let's go for it. So here we are in the last slide. We have finished. I hope you enjoy. Let's see what we saw in this video. Okay, there were only three steps. There are only three steps. Okay, so let's go for the step number one. Let's remember it. It was just thaw. The expression supernatant, well, in case it was frozen. Okay, then we centrifuge it, and after centrifuging, we incubate it with benzonase and magnesium chloride for 30 minutes. Remember, with the benzonase and the magnesium chloride, we get rid of the DNA from the mammalian cells. Okay, and afterwards we incubate it with detergent for another 30 minutes. That was all. With this, we get rid of a lot of contaminants. Okay, the second step, what was it? Was preparing the machine, preparing the TFF device, and we prepared TFF buffer. And after preparing the TFF buffer, we just equilibrate the TFF device with the buffer. That is to say, we leave the TFF machine in the best conditions to receive the sample. And what was the third step? It was concentrating, cleaning, and buffer exchanging the sample with AAV-OSK. And the only thing that we do is setting up the machine and yeah, turn it on the machine. It did everything. It concentrated, it cleaned it, and it buffer exchanged. Okay, it was very, very easy. So as I have said, an easy technique, very easy to perform. I hope you enjoyed very much. Yay! We have the protein super, super, super clean, almost ready. Well, there are a couple of steps more that are needed, okay, in order to purify the protein properly. At that point, the protein is very, very, very clean, close to 95%, sometimes 99%. So we require another step in the purification process, okay, but we will see it in the following video. Thank you very much for your company. Thank you very much for watching the video. Big thank you is saying Mr. Novosumer. Okay, as I have told you, the working instruction related to this video will be available in the following months in www.novosumer.com. The website will be released soon, very, very soon. And this working instruction that is going to be available in novosumer.com is compliant with CGMP, ISO 9001, ISO 45001, and ISO 14001. These, both of them, are standards for quality. The ISO 41001 is the standard for health and safety, and ISO 14001 is the standard for environmental. Okay, to respect the environment, which is important. So, my friends, thank you very much. I hope you have enjoyed. I hope I see you in the following videos. Okay, subscribe to the video or subscribe to the channel or press like if you liked it and enjoy. Enjoy it very much. Thank you very much. Bye bye.